So we're getting into our next body system, which is the muscular system. All right, and so from anatomy, you remember you had to know all these different muscles, all these different types, and were they antagonistic, were they synergistic, all that kind of stuff. So some of this will be a little bit of review like when we talk about muscle types, but really what we're going to focus on more than anything is how does the muscle actually contract, right? That's what our focus is. So let's go ahead and with this video get into just different types of muscle cells, which is a little bit of review from histology. We'll talk about the features of each and then move on from there in other videos. So if we think about muscles overall, what is their general function? What's the purpose of muscle? Some of the things that are more obvious are like producing body movements, being able to walk, being able to talk, being able to run, right? Things like that. Some of the things that are maybe not as obvious or not as evident are things like stabilizing your body positions, being able to stand in an upright posture. There's little tiny changes, little minute changes that are made, right? Little tiny changes in muscle contraction that allow you to stay upright, right? Even if you were to walk like on uneven ground. And then as we get to the less obvious ones, we get to being able to move substances around within the body. So the heart, that pumps the blood. Arteries, moving blood, arteries and veins, they all have muscle as well as in the GI tract to move food and fecal matter along throughout the GI tract so that we can move things along to absorb nutrients and then eliminate any waste. And then lastly, generating heat. We will contract muscles when we shiver, and that shivering is going to use energy, but it also produces heat as like a byproduct of that muscle contraction. So no matter what type of muscle we're looking at, they're going to have all of these particular properties or characteristics, right? So number one is contractility. This is the ability of the muscle to shorten, right? It's going to move in towards the middle of itself, right? So it's contracting inward when it's stimulated, shortening the entire length of the muscle. Number two is excitability. This is the ability to respond to a stimulus because it's going to depolarize its plasma membrane. We're going to talk about the neuromuscular junction and how that is going to trigger that excitability. Extensibility, this is just the ability to stretch outwards without being damaged, so it's extendable. It can extend out past its normal length, so without having to, fix any damage or anything like that. And then elasticity, once we've extended, we can snap back to our normal length after being that stretched out. So if we think of it all together, right, muscles are excitable cells that in response to a stimulus will either contract or extend, and that regardless of contraction and extension, they can snap back because they're elastic, they can snap back to their normal length when not being stimulated. And we have three different types of a muscle that we can categorize them into: smooth muscle, cardiac muscle, and skeletal muscle. So let's talk about skeletal muscle first. Skeletal muscle is pretty much just attached to your bones. They're long, they're lean fibers, so they're very cylindrical, they're very uniform in their appearance. We see that these guys have a lot of nuclei in them. So if we look at the picture here, right, we see like this is one single cell. So when we say a muscle fiber, we're really saying a muscle cell. So it's one single cell, and yet we have lots of nuclei all the way down the line. There's no branching, so they're long and lean and thin, and they have these striations. And the striations are these stripes. In this case, the cells are running left to right, and the striations are running up and down. We'll talk about what those striations are in a little bit. Now, when it comes to the control of skeletal muscle, this is voluntary. It's somatic motor control for this. So you have conscious awareness and conscious control over these muscles, and that we're organized into these units called sarcomeres. Sarcomeres are going to have two different proteins called actin and myosin, which are going to help with the actual contraction itself. So the actin and myosin are the ones that are actually allowing causing the muscle fiber to shorten. The regulatory proteins are troponin and tropomyosin. Basically, what they're doing is they're going to be helping regulate when the sarcomere contracts. So they're involved with kind of in a way you think of it like them turning on and off the muscle. We're going to contract in response to the neurotransmitter acetylcholine. We have a lot of sarcoplasmic reticulum, and within that we have lots of calcium. And these contractions are very, very fast. We don't see junctions between these muscles. When we look at cardiac muscle, we're only finding this in the heart myocardium, which is the muscle layer of the heart. They are kind of similar to skeletal muscle and they're long and cylindrical. However, some things that make them a little different is there's only one nucleus found in the center. We have branching that occurs, and then we do have junctions between each cell where we find the intercalated discs, which is where we find gap junctions. And those gap junctions are going to be used for rapid control. Like skeletal muscle, they are also striated. So if we look here, right, at this picture, what do we see? So we see kind of long and lean and cylindrical. That's similar to skeletal, but we see branching, right? We see that it's more branched. It's not all just kind of going in one direction. We do see the striations that they share with skeletal, but then we see a couple of striations that look darker than the rest, and those are the intercalated discs where we have our gap junctions. We're also organized into sarcomeres, right, with the actin and the myosin being the contracting proteins. Same thing, troponin and tropomyosin are going to be the regulatory proteins. However, rather than acetylcholine, we're responding to norepinephrine. And you'll notice that we're responding to norepinephrine coming from the autonomic nervous system, meaning this is also going to be involuntary, right? Meaning we don't have conscious control. We have some sarcoplasmic reticulum, and we use that to store our calcium source. We also have the interstitial fluid for calcium as well. The contraction rate is a little bit slower. It's moderate now. And we do have those junctions. I mentioned them just briefly before talking about the gap junctions. We'll get more into that kind of stuff later. But for right now, just kind of remember they have these gap junctions. And then smooth muscle. Smooth muscle is really different from the rest. We notice it looks very different, right? It looks very different in comparison. And we find it in, the walls of the viscera. We find this basically in all the other, like the guts, right? So in your blood vessels, in the ciliary bodies of your eye, in your reproductive tract, lining your digestive system, the bladder, right? All those types of things are going to use smooth muscle. And they're going to be much shorter. They're spindle shaped, so they're almost kind of like, they kind of have a nice little belly to the cell, and then they kind of point out on the ends, one single nucleus, and we notice they're smooth in appearance, meaning there are no striations. This is also controlled by the autonomic nervous system, and so this is involuntary. We do not use sarcomeres anymore. We are using actin and myosin, but not sarcomeres. And so that means since we're not using sarcomeres, we can change up the regulatory proteins. In this case, calmodulin and myosin light chain kinase are the ones regulating that. They're responding to norepinephrine from the autonomic nervous system. We don't have a lot of sarcoplasmic reticulum, although it still is our source of calcium, as well as the interstitial fluid. And the contraction speed is slower. We do see junctions between them as well. However, we don't see them as obvious like the intercalated discs we saw with the cardiac muscle. So we don't get this nice like sarcomere, that sarcomere structure. And so that's why we are smooth, because the sarcomere is what gives these striations to cardiac and skeletal muscle. And so rather we just see like what's called a dense body, which is, see these little points almost look like little beads there. And then we are stretching those myofilaments, right, like the actin and the myosin, from dense body to dense body, right? So it almost looks like someone did like some quilting, and then the myofilaments are running from one like knitted down point to another. They're really similar in terms of function to the Z discs of a sarcomere, which we'll talk about in a little bit. But because of the change in how they're designed, their contractions do start more slowly, but they last longer. Right, and so that's a little different. They also can shorten and stretch more than skeletal and cardiac. So they're a bit more flexible in terms of their movement, but there's a trade-off. They have to be a little bit slower with their movement and their contraction. Now we can be a single unit or a multi-unit fiber, right? And so single unit right here, we see there's a pattern that allows the individual fibers to contract. So like in the iris, the erector pili for like that attached to your little hairs on like your arm, in your blood vessels, whereas multi-unit innervation, we have a lot of different neurons that are linking up with the smooth muscle, and so multiple muscle fibers can contract as one for more complex movements like peristalsis. So we see like single unit is good for small things, simple things, like we just want to stand a hair on its end, versus we want to move all this food through the gut. And so here's just a quick summary. I'm not going to read this to you because that's kind of silly, but just take some time, maybe pause here for a little bit, read through this, and honestly make your own summaries of comparing skeletal, cardiac, and smooth muscle. We will go over some of the basics in lab. However, you really should make some diagrams for yourself on this, looking at the different muscle types.